Good morning. So today I will be discussing about the lactic acid production. Firstly, there are certain prerequisites that are required for the industrially microbiological processes, and these involves three prerequisites. Prerequisites are basically the pre-requirements, the requirements that are required before carrying out the fermentation process or any industrial process, that is the large scale production of any product. So the first prerequisite in the industrial microbiological processes is the organism. The first requirement is the organism. The choice of the organism depends upon the type of product we want to produce. That is, the organism, it needs to be stable. The characteristics involves that it needs to be stable. The microorganism which we are using for the production of any economically valuable compound, it must be stable. The stability is the major important factor. Then after stability, it's pre-requirement treatment. Prior treatment required before the large scale process. Then next, after the stability and the requirement of the treatment, the next is that it must be easily available in the environment. It must be easily available. And the last main important point is that it must be non-pathogenic. The microorganisms that we are using in the culturing the product is the non-pathogenicity. The microorganisms used is they must be non-pathogenic. Otherwise, they would create the disturbance in the production of the product, and they would retard the fermentation or any other large scale process. So the last point is the non-pathogenicity of the microorganism. The next prerequisites in the industrially microbiological process is the medium. There are certain medium depending on the requirements for the production of the product, like the whey is the dairy waste product for producing lactic acid. Whey, it is the dairy waste product. The characteristics that my medium must possess is that it must be easily available and readily available in the market, and it must be, the next point is the cost. It must be cheaply available. It must be cheap. The raw material that we are using for the production of the commercially important compound is its cost. It must be cheaply available. Then the last prerequisites in the industrial microbiological process is the product, the end product. Generally, the large scale fermentation or any process, the large scale process is carried out in 5,000 to 50,000 of gallons vessel. The capacity ranges between the 5,000 to 50,000 gallons, that is the culture vessel or the fermenters, or we can say the bioreactors. During the large scale up process, after the large scale up process, the recovery and the purification is the major step involved. Recovery and the purification of the product is the crucial step in the production of any product. Because in the end the product it must be free of impurities and contamination. So the purification and the recovery is the major step involved in the production of any product. Then in case of lactic acid production, the organism required, firstly, major prerequisites in the development of a lactic acid is the organism. There are certain organisms which are used in the production of lactic acid. So the major important bacteria that plays role is the Lactobacillus bulgaricus. So Lactobacillus bulgaricus is the major producing microorganism of lactic acid, and apart from Lactobacillus bulgaricus, there are certain other bacteria also that play a major role in the production of lactic acid, and they are like Lactobacillus casei, then Lactobacillus delbrueckii, Lactobacillus psychophilus and Lactis species. So the apart from Lactobacillus bulgaricus, the other microorganism, that is the bacteria, they are Lactobacillus delbrueckii and Lactobacillus casei. So mainly Lactis species. So the major lactic acid producing microorganism is the bacteria. Other than the bacteria, that is apart from bacteria, the fungus is the microorganism that is also utilized in the production of lactic acid, and mainly in particularly Rhizopus species. In case of fungus, Rhizopus species is used in the economically valuable component in the production of the lactic acid, fungus, and in particularly fungus, Rhizopus species is selected for the production of lactic acid. Moreover, in Rhizopus species, the major fungus that is used in the production of lactic acid is Rhizopus oryza. So the Rhizopus oryza, it is also used nowadays in the production of lactic acid, but it has got certain disadvantages and demerits. Like first is it has got very low production rate. And this low production rate is most probably because of the production of byproducts. It causes the production of certain byproducts like acetone, ethanol and formic acid, due to which the production rate becomes very low, and so it is the demerit of using fungus as a microorganism in the production of lactic acid. So generally we do not prefer to use this Rhizopus species because of its low production rate, most probably due to the formation of certain byproducts like formic acid and ethanol. So because of the production of these byproducts, the rate of the production of lactic acid decreases. So that's why we don't consider to use it as a microorganism for producing lactic acid. Second major prerequisite, as we have studied earlier, is the medium. So there are certain raw materials which are used for carrying out the lactic acid production. The certain raw materials include corn, molasses. Molasses is the waste product of sugarcane industry. Other raw materials involved are wheat, rice, barley, sweet sorghum, wood, sweet sorghum, rice, barley and potato starch. And the major lactic acid producing raw material is whey, which is an dairy product. As I have already discussed about whey, so the major step involved in the lactic acid production is the hydrolyzation of starch into glucose. The major process is that the starch must first be hydrolyzed to glucose and then to lactic acid. So the carbohydrates, they play a major role in the production of lactic acid. So generally we prefer to use carbohydrate containing raw materials, because carbohydrates are the major nitrogenous compounds and they are used in the production of lactic acid. So the carbohydrates containing substances are generally used as raw materials, as I have discussed earlier, like molasses, potato starch, corn, wheat, barley, rice. So these all are raw material which are used in the lactic acid production, and the major lactic acid production bacteria is the raw material is whey. So we'll talk about the process of lactic acid production by using raw material whey. So whey is a dairy product waste, and it is so people thought of using it as a in making useful product. Earlier the whey was treated, it is generally released from the industries as untreated. It is usually in untreated form, and it requires certain treatments before we use it for the lactic acid production. So because from the standpoint of certain pollution problems as well as for economic reasons, it is desirable to use whey for in making certain useful products. So we started using it as a raw material in the production of lactic acid. Now whey as a raw material, since whey is very high in carbohydrate content, it has got very high carbohydrate content, so it is used in lactic acid production because of its high carbohydrate content. And the carbohydrate, it is present in the form of lactose in whey. So it is present in lactose form, which is further hydrolyzed into glucose and then into lactic acid. So here lactose is converted into glucose and then it is further hydrolyzed to the lactic acid. The IUPAC name of lactic acid is 2-hydroxypropanoic acid. And its chemical formula, the chemical formula for lactic acid is CH3CHOHCOOH. After the medium, we come to the product, that is the fermentation process. So firstly the stock culture is prepared in the skim milk, and skim milk is basically the whole milk that is free from the fat, milk fat. So generally the medium used for stock culture, for preparing the stock culture, is skim milk, and it is usually fat free milk. So all the extra fat from the whole milk has been removed, so that is skim milk, and it is used to maintain the inoculum. So in order to produce the inoculum, generally skim milk is used, and milk is the most appropriate and the suitable media. To prepare a sufficient amount of inoculum, the stock culture, it involves the addition of the inoculum to the main fermentation tank. Then after this, it is successfully transferred and cultured firstly into the sterile milk, firstly sterile milk, then after that it is transferred and incubated into the pasteurized milk. Pasteurized milk, and then further lastly the whey culture. Then from the inoculum, from the whey culture, it is transferred into the fermentation vessel for the fermentation, to carry out the fermentation process, and it is added in the volume, that is, it is added is 5 to 10% volume of the product that is to be fermented. So 5 to 10% of volume it is added to the fermentation tank. Generally milk is used to build up the inoculum. Major medium used is milk for building up the inoculum. The incubation temperature that is set is 43 degrees Celsius. 43 degrees Celsius is the optimum temperature that is used for the incubation, and it is set because in order to favor the growth of Lactobacillus bulgaricus and to inhibit the growth of certain other bacteria that might grow in the culture vessel. Then after, the next process is the fermentation. So during fermentation a slurry of lime is added into the culture vessel, that is CaOH2 slurry. A slurry of lime is added to the culture vessel irregularly. It is not continuously supplied to the vessel, rather it is discontinuously applied to the culture vessel in order to neutralize the acidic conditions that are maintained in the culture vessel. So in order to neutralize the acid, lime slurry, that is lime slurry, is added, and then after that calcium lactate is produced, that act as a calcium source in the culture vessel. So otherwise accumulation of the acid there, it would retard the fermentation process. Upon the completion of the fermentation, after and it generally takes 2 to 3 days. It generally take 2 to 3 days for the fermentation process to get completed, and after which the material that is left in the tank, it is boiled. It is boiled to coagulate the proteins which are present in the culture vessel, after which filtrate is left. So after that the filtrate is evaporated in order to remove the water under vacuum. So next step is the evaporation step, in order to remove all the water under vapor. Then the next step is the purification step. After the filtration, purification is done in order to get the purified form of the lactic acid, the end product, that is lactic acid. So the major biochemical reaction that is taking place during the fermentation is as follows. The reaction in biochemical reaction that is carried out by the microorganism Lactobacillus bulgaricus. Lactose, after the treatment of lactase enzyme, is getting converted into glucose and then to lactic acid. Lastly, the applications of lactic acid is first cosmetic. In cosmetic industry it is used as anti-acne agent and in whitening agent, and it is also used as humectant, that is the moisturizers. In pharmaceutical industry it is used in dialysis solution, in mineralization solutions, and the last application is the food industry. In food industry it is used as a preservatives and flavoring agents. Thank you.